redness and heat on the face is marked. So, in conclusion, homeopathic medicines are very much effective in curing angioedema. No conflict of interest, no funding, Mr. Bibliography. And I thank each and everyone for giving me an opportunity to present, especially Sushi sir. Thank you all. And on the
Thank you so much, ma'am. Our next participant is Sayed Khalida Izhar.
Yes, sir. Good. Sir. Hello, sir. It's visible.
Yes, please.
Sir, it's visible.
No, not. No.
Sir, it's
Next
visible.
participant is Swetal Patel. Be ready. Next one is Fojia Chaudhary.
Sir, it's visible.
Yes, visible.
Yes, good afternoon, all of you. My name is Sayed Khalida Azhar. Today I am presenting my research work. I am from Integral University, Lucknow. My title is Isolation, Identification and Characterization of Actinobacteria and the Antimicrobial Activity against Pathogenic Strain. Abstract: Actinobacteria are gram-positive bacteria that make spores and have a high GC content. These are a class of bacteria that have received the least attention but are widely known for producing secondary metabolites. The objective of this research was to study the antimicrobial activity of actinobacteria isolated from soil samples taken from different locations of Lucknow, Uttar Pradesh, India. Yeast extract, malt extracts, and starch casein agar and glycerol aspergillus agar are the specific actinobacteria isolating media that were used to study and include 56 strains that had been isolated from the soil. A total of 115 microbial colonies was screened and 6 positive actinobacteria were identified using 16S RNA sequencing after being selected based on their morphological, physiological, and biochemical properties. These actinobacteria were then tested for their antimicrobial properties using the perpendicular staining method and the Kirby-Burger method, respectively at the primary and secondary screening levels. Isolates SKIX, SKIH, and SKYR shows exhibiting antimicrobial activity against Asorius fumigatus niger. Thus, from the present study, it can be concluded that actinobacteria isolated from soil might be a valuable resources from the development both spectrum antimicrobes. Introduction: Actinobacteria are a group of positive bacteria which shares the characteristic of both bacteria and fungi. Interesting application of actinobacteria is the use of their secondary metabolites as bioherbicides against weeds. Approximately 10,000 bioactive metabolites are produced by actinobacteria. Some actinobacteria are also animal and plant pathogens such as Microbacterium, Corynebacterium, Nocardia, Rhodococcus, and a few species of Streptomyces. These species are gram-positive filamentous bacteria which produce aerial hyphae and differentiation into chain of spores. These secondary metabolites present an upland source of biological active compounds such as antibiotics, agrochemicals, enzyme, immunosuppressant, antibacterial, and anti-cancer agent. Thus, concerns on the environment production made it essential to develop safe, non-toxic, and eco-friendly bioherbicide in order to decrease the chemical input into the ecosystem. Methodology: soil sample collection and pre-treatment, isolation of actinobacteria from soils, different sample. Then I am morphological characterization of actinobacteria. First of all, primary antimicrobial screening by perpendicular stick method, and then secondary antimicrobial screening by well diffusion method. Then IM characterization of 16S RNA gene sequencing, then biochemical analysis, a production of secondary metabolites. First all of secondary metabolites production from the antibacteria was performed the submerged stage fermentation, and this methodology of secondary metabolites production using a separation funnel, the upper layer solvent phase containing bacteria compound was collected. Then rotary vacuum evaporation at water bar temperature of 45 centigrade, and then 200 rpm to eliminate to the solvent and obtain the extract in crude form. The obtained crude extract was evaluated and mixed with 1 ml methanol to from the stock solution and was stored for further. The active compound produced by both strains of actinobacteria are the characterized by molecular technique like GCS-MS. Uh, First of all, I am presenting the diagram showing the results. Collection of soil sample, then pre-treatment after incubation of 6 days. Pure colonies are found. Primary antibacterial screening and antifungal screening. Then secondary antibacterial screening after molecular characterization of 16S RNA sequencing, then GCMS. Growth of actinobacteria on different agar medium. Isolation actinobacteria strain positives. Actinobacteria isolated on actinomycetes isolation agar, and actinobacteria isolates on star cassin agar. My results: positive isolate of actinobacteria from soil sample of different location: Gomti Nagar, Chinnahat and Basti region, Siddharth Nagar region, and Integral University. Morphological characteristic of actinobacteria showing results. Then good, moderate, and weak. No, some are they produce no activity. I am correct, morphological correcting for growth, color, pigmentation, elevation, and surface margin. Primary actinobacteria screening of actinobacteria. Some pathogenic strain for using E. coli, Pieraginosa, Asorius, and K. Capsulina pneumonia. They produce results. Secondary antibacterial screening of actinobacteria, some pathogenic strain against Saboni, E. coli, K. pneumonia, Bacillus aeruginosa, and Asorius. Some actinobacteria give best results like SKIH and XKY. Antifungal screening of actinobacteria, some fungus against actinobacteria, Trichoderma, A. fumigates, A. flavus, and in Niger. Biochemical tests of selected actinobacteria strain give results. Indole test, methyl red test, Bacchus-Prosca test, catalase test, starch hydrolysis, casein hydrolysis, modality test, and carbon hydrate fermentation urease test. Then pictures of positive strains. Molecular characterization of the selected isolate using 16S RNA sequence amplification. The identify of the selected 6 isolate on the basis of their antimicrobial activity was confirmed using 16S RNA sequencing amplification. Table 1 presented. I am using the BLAST tool in the gene bank database. Has been concluded that the 2 isolate belong to actinobacteria. Further, the phylogenetic analysis confirmed that is isolate belong to the Microbacterium SKIX and Microbacterium SKIH genera. The partial 16S RNA gene sequence of the isolate SKIX and SKIH has been deposited in the NCBI gene bank with the accession number OP071245. 16S RNA amplification of isolated actinobacterial strain SKIH Microbacterium species, Microbacterium hydrothermal, and SKIH Microbacterium trichosanthelium Microbacterium species. They are two presented actinobacteria, a phylogenetic tree of SKIH and phylogenetic tree of SKIX. Production of secondary metabolites. First of all, I am using ISP2 media prepare after 10 days for incubation and then 25 days of incubation. Extraction of secondary metabolite by liquid extraction method in a separating funnel, crude extract formation, then extract secondary metabolite. In conclusion, the newly isolated restrained microbacterium species SKIX and SKIS showed broad antimicrobial spectra as it inhibit gram-positive bacteria, gram-negative bacteria as well as fungi. These significant results make the strain suitable for further investigation and still exploration. Actinobacteria can produce valuable secondary metabolites, including bioactive herbicidal compounds that might be used to create a good candidate for biological weed control. Reference. Thank you so much.
Thank you, Said Khalida, for nice presentation and research work.
Okay, sir.
Yeah.
Thank you so much.
So there are several other earlier works on the same line has been performed by several microbiologists. What's
Yes,
the
sir.
no in comparison to that in the current study performed by you? Yeah. Could you may explain 1 to 2 point of the novelty of your work?
Sorry, sir. I'm not listening. Please explain. Repeat
What is the
question.
what is the nobility of work? Because several earlier schools of microbiology has been worked upon the same line of microbiota, but what's the
Yes, sir.
nobility of your work?
Sir, my work for I am research for herbicidal activity for actinobacteria. Then herbicides. I'm एक्टिनोमाइसिटी से हम हर्बिसाइडल प्रोडक्शन करना चाह रहे हैं कि जो
Yes,
हमारे इन्वायरमेंट को जो सस्टेनेबल और प्रोटेक्ट करे, टॉक्सिसिटी उसकी नॉन टॉक्सिसिटी हो, जो इतनी ज्यादा मैन्योर ये सारी चीजें हो रही हैं जिससे हमारा जो यील्ड हो रहा है क्रॉप की उसकी उसको लो करने के लिए ये हमारे वर्क की नॉबिलिटी रहेगी.
yes. But in the current presentation, you don't have applied these kind of
जी
microbes
जी
over the herbs.
जी सर,
And
जी सर. आई एम
it means,
गारंटिंग सर, वो मेरा रिसर्च वर्क है जो आगे चल के है.
okay,
अभी हमने जो जी जी सर, जो
नहीं,
अभी रिसेंट में मतलब हमने सर जो पार्ट दिखाया इसकी नॉबिलिटी जो है, जो मेरी माइक्रोबियम स्पीसीज है
हम्म,
SKIX एंड
हम्म,
SKIX, उसमें
yes,
सर जो इसकी एंटी माइक्रोबियल हमने अदर्स और जो अभी हमने पेपर्स पढ़े हैं उससे
जी
ज्यादा
जी.
ये इनहिबिट कर रहा है उसको.
Yeah, it means it is more efficient than the earlier reported fauna.
जी.
Yeah.
यस.
Okay. Second thing, you have used for the molecular characterization
यस.
16 sRNA. Okay.
Yes, yes, yes.
And there are several other conserved genes are like 5.5 and 5 sRNA is also present in prokaryotes that were used. Why you have not chosen that? Why
सर
chosen 16s?
पर 16 ईयर्स इसलिए कि जो एक्टिनोबैक्टीरिया होती है स्पीसीज
जी.
उसके लिए 16 ईयर्स ही पर्टिकुलर है सीक्वेंसिंग के लिए.
And the primers designed by yourself or you have taken an universal primer?
सर माई सेल्फ प्राइमर
Okay, okay.
चूज बाय मी.
It means you are efficient in the biomolecular work at the level of primer designing to the application. Yes.
यस 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 सर.
Thank you, Khalida, for your nice presentation.
थैंक
Thank
यू
you
सो
so much.
मच. थैंक यू सो मच सर.
Our next presentation by Dr. Anand Kumar Thakur sir.
Namaskar. Thank you.
Thank you sir.
Thank you. Thank you. And I have to share my PPT. Just a minute. Is it visible?
Yes, sir, visible.
Fine, fine. First of all, thank you, Dr. Siddharthan, for
Thank you,
giving
sir.
me this opportunity to share some view of the integrated pest management of a project which is just ongoing. Uh, means in October I received this project. So the very primary work is going on right now. So as you all know that beetle gourd, Moverantica carencia, is a very common vegetable. It's very popular in southern part of the India, but it is used by the farmers of every part, especially northern and eastern region of western region also of India, and it is known for its medicinal value. So this project has been given for the Jharkhand purposes to just know that what kind of challenges are facing means farmers are facing the challenges uh, to cultivate the bitter gourd, and how integrated pest management can be applied in Jharkhand especially. So as we all know that this is a very popular vegetable, means because the taste is not so good, we all know it, but its medical impact is always so much that we prefer this particular vegetable on our table, uh, and we eat it to get good health benefits. Children never like this bitter gourd. Even many of the adults also deny to eat it because of its taste. But when we detail them, its medicinal value, especially in the field of blood purification and to decrease the blood glucose level, or to decrease the lipids in body, then all the time. Juice of bitter gourd is also very common nowadays, and we can know that its use is so much in Ayurveda and naturopathy. Even yoga Guruji Ramdev they all he always promote the juice of Karela for an anti-diabetic medicine. So farmers has to cultivate it without any big problem, and develop its market and its utility in both as a vegetable and as well as natural drug also. So project is just in a very initial process. So it is just a kind of sharing of the informations related to the beetles of this bitter gourd. It is widely distributed in Asia, East Africa, and South America. In India, the southern and northern parts are main cultivators. Medicinal value, as I told you, that it has hypoglycemic property. Sorry, it is incorrect. Rather, it is a hypoglycemic. It should be okay. So it is known for its clinical value to decrease the blood glucose level. Any person who is suffering from diabetes mellitus use this particular vegetable to just make a balance of sugar in his or her body. It has been reported by Singh that a stimulatory action on beta cells. It was treated on mice, and he reported that after the treatment of this particular, the secondary metabolite of this vegetable, the islet of Langerhans activated and beta cells started secretion of the insulin. It reduces adiposity. So we know that in our society, people are very concerned about their fat, and they want to just reduce it as per the BMI. So it is helpful, and so the recent reports are just trying to establish its medical the people who are suffering from this kind of adiposity. Now, these are some beetles we are cultivating in a very natural farmers. We have make some agreement with the farmers just near the Ranchi, and they are cultivating um, Momordica carantia, and our team regularly visit in 1 day, 2 day. We go there and collect the samples, and the farmers, they are cultivating this uh, vegetable as per our guidelines. So we are providing each and every facilities to them, okay, and the financial support also to cultivate this Momordica carantia. We are using right now. We are actually using the wild plant only. Okay, so we have to standardize it in our first life cycle of that Momordica carantia to establish its standardization in the specific climatic conditions of Jharkhand. If you are from Ranchi, then you may know that it is a very good place for the purpose to cultivate fruits and vegetables and now flowers also. So we have reported 6 beetles. Okay, so these beetles are actually very common. We are reporting it, and we are getting the exact data and its life cycle. That Raphidopalpa, it is means what I can say, that this beetle actually affect the leaf and the young branches of that wild type of Momordica carantia, and very rapidly it means destroys the young leaf and young branches. And with overnight, we have reported that the leaves and branches was completely vanished. Only veins were there, and leaf was completely eaten by the Raphidopalpa. Second one is Olecaphora formalis. It is also very commonly found. Okay, and in ICR plant here at Ranchi, they are also doing some work on beetles. So we are in regular contact with them. This is the third one, and that is called Olacophora nigripenis. So it is very little. The presence of this particular beetle is very less compared to the means pink whip beetle or hadda beetles. But they are also been found a very small amount, a small number rather. The fourth one is Epilacna. This is very common. This one is very common. Okay. So I could not get time to just put my photographs here. So I took it from net, and this is very common and very prevalent. During rainy, what happened that rain was a little bit in November till first week of the November. So October, November, there was a good rain. And we had observed that during that period, this is Epilanca. There was 2 species of this Epilanca, and they were very prevalent. They were just doing all the means damage to the leaves, young leaves especially. Continuously they were damaging. They were reproducing and producing eggs and larvae. We were just getting photographs of all these things. And yeah, this one is also. So Vosdu Valley, it was also, but comparative to this particular species, this admirabilis was very common. So it was the common among the all 6 types of beetles what we observed there. And Epilanca, uh, it was also observed in some cases. So still we are very much concerned about the types, because we have to collect it from different plants, haze of the plants, because the farming pattern of this particular vegetable is a very common, that they make some roofy structure of bamboos and young plants, and just spread the plants on that particular, and the vegetables just hang there in the. So what kind of damage actually they are doing? So both nymph and adult insect were damaging the crop. Mm. And especially the nymph, means the larval stage. The nymph was very much voracious, continuously eating the leaves. Means we have also determined the size, the length of the leaf, this area of the leaf, and the rate of just feeding of the beetle on that leaf. It is also means calculated. Now, what have to do? We have to just develop an integrated pest management model. Okay, and this model is also taking the help of uh, thermohygroscopic changes, means changes based on the temperature and the changes based on the humidity. And we have to develop a data, and we have then the final stage we have to just to use this data through two-sex method to standardize its means simulation. It's a simulation software. We have to develop some data, and we have to establish that what level of beetle can harm if the temperature and humidity will change in the agricultural area, especially in Jharkhand. So it is also part of that project. So the effect of pest can be reduced. We have to just do these things. So we have these are actually things that we are actually just training for the farmers. So they have to do these things, okay, and we have to put the record on in a proper way. As we all know that the life cycle of beetle is having eggs. This is very common photograph. Just besides the leaf, we gather these eggs and larvae. And grub is the larval stage, which is just like it's a very good one. It appears like a fruit of some plant which actually stick with our chiri plant, like just like that. That's the thing. So it has a bristles on the body, and but through this bristles, it intact with the uh, leaf part and the young branches part, and they start feeding as per their demands. And pupa, it is uh, oxalate beetle type, octyl type, and uh, they means after for 5 to 8 days, they just emerges into an adult. So this is the basic life cycle of that. Yeah. Now, what our strategies are to look after these things? First, the first part of the phase is just to develop their interactions with what types of beetles are there, how farmers are getting problems from these beetles, what type of life cycles of these beetles are there, and in what stages they are damaging the crop, what extent they are damaging the crop, means that damaging the vegetables, and what is the impact of the production of that vegetables, how much it is reduced. So this time we are not using any type of insecticides or pesticides to kill them. It is wild, no any kind of means chemicals are used there. Even we have also treated soil in a very proper way, so that the first is we can standardize the wild type one, and we can understand that level of infestation and damages just doing by damages by these beetles. It is very primary work we are doing right now. So collection and destruction of the dwarf fruits and infested fruits on the plant helps in the management of the fruit flies also. We are just looking out that what kind of damages, what type of other insects or some other predators who are killing these beetles. We are also looking forward them, and then we will start the spraying of some botanicals, secondary metabolites, and other second types of chemicals that we will prepare, and we will see the effect of these on the life cycle of uh, beetle. So then we have to move through. We have to just check the effect on the egg. Then we have to check the effect of these secondary metabolites on grubs, and then on pupa, and then on adult. And then we have to develop a standardized pattern of their infestation, and then only we can get the exact result. It will take another 1.5 years to get the result. Thank you very much. So it was just a primary information about my project to all of you. Thank you for your patience listening. If you want to ask anything, please go ahead.
Okay. So the next presentation would be from Shwetal Patel. Ms. Shwetal?
Sharing my screen. Hey, is it visible to all? Is my screen
Okay, it's visible.
Good afternoon, everyone. Myself Shwetal Patel, and I'm going to present my research on effect of processing methods on bioactive compounds, nutritional quality, and toxicity of Mucuna pruriens seeds. Mucuna, it is an underutilized crop plant, and it is mainly used to treat the Parkinson. But in spite of its potential to treat the Parkinson, it is also having high protein content. But because of the anti-nutrient compounds, not anti-nutrient, we can say the bioactive compounds present in the Mucuna seeds, they are causing certain health issues. And that's why the utilization of Mucuna beans as a protein source is very, very limited. Okay, since Ayurvedic time, it has been used to prepare certain medicines, okay, because of the L-DOPA. The major constituent of Mucuna pruriens seed is L-DOPA, and it having neuroprotective activity. But our aim is slightly different. We are exploring the potential of Mucuna bean as a protein diet. Because it is not only used by the tribal people, but it is also used to prepare the animal feed, and it has potential to increase the fat content or in animals, mainly in chicken. So our study has been divided into two different groups. One is removal of anti-nutrient compounds from Mucuna seeds, and second its utilization as a protein diet. Because we are using certain processing methods. We are combining the traditional processing method as well as we have employed certain new processing methods. And in our study, we have treated the Mucuna seeds with frying, second roasting, then overnight soaking in cow dung, cow urine, boiling with 1% NaCl for 1 hour, and overnight soaking in milk, buttermilk, and we have also used the sprouting method, that is our old traditional method. And then after treatment, the seeds are then dehulled and dried, and grind into the powder, and that powder is further utilized for the analysis of anti-nutrient compounds. And the anti-nutrient compounds includes L-DOPA, the major anti-nutrient compound present in Mucuna pruriens, and this L-DOPA can cause the nausea, vomiting, and sometimes the dysgeusia also. And the another compound is the phenolic and flavonoids compounds. We can call them as a bioactive compounds because they have certain health benefits. Then another one is phytic acid and the oxalic acid, two major anti-nutrient compounds. And all the bioactive compounds are measured by using standard methods, and the DPPH free radical scavenging activity to analyze the potential of Mucuna seeds to scavenge a free radical was measured by the method described by Ebodu et al. Further, The nutrient compounds, the major two nutrient compounds such as protein and carbohydrate were measured using AOSA method, and protein content was determined using the Gerdau apparatus. After the analysis of nutrients, we have selected the overnight soaked seeds in milk as our potent protein source, and we have employed the treated and untreated seeds on the rat model. And the toxicity study was carried out using the albino rat model as per the OECD guideline 425. All data obtained were further analyzed using SPSS software. Results and discussion. As you can see that the processing methods are significantly reducing the amount of bioactive compounds. The major limiting factor, that is phenolics, was leached out by overnight soaked seeds in milk. And the second major anti-nutrient compound, tannin, was also found to be in least amount in the same treatment. Phytic acid was further reduced by the autoclaved seeds, and the L-DOPA, the major limiting factor was also leached out by the overnight treated seeds in milk. So we have decided that the overnight soaking in milk treatment was found to be the more effective, as it results in elimination of phenolics, tannin, L-DOPA, and phytic acid. Not only the leaching of anti-nutrient compounds, but the conservation of nutrient compound is also necessary for the utilization of seeds. So we have carried out the carbohydrate and protein analysis using the standard method, and it has been shown that the sprouted seeds conserve the protein amount up to 32.90%, and our raw seeds, the protein content in our raw seed was 33.03. So only 0.4% difference was found, and the amount of carbohydrate was also increased up to 34.55, followed by the seeds overnight soaked in milk, and it has conserved the protein amount up to 20%. Plus the carbohydrate amount was also increased up to 42.34. But the major problem with beans is that the mucuna seeds, due to their larger size, it takes time in sprouting. It takes 2 to 4 days for sprouting, as well as start the fungal contamination because of their carbohydrate and protein content. So we have selected the overnight treated seeds in milk for our further study. And the toxicity assessment was carried out using the albino rat model by following the guideline number 4, OECD guideline number 425. Single dose of 2000 mg per kg was given to the albino rat, healthy albino rat, both as male as well as female, and they have divided into 5 groups. And after 14 days, the rat was sacrificed, and the major organs, vital organs, were isolated and further used for histopathology study. And the blood was collected by using retroorbital plexus, and it was used for the hematological parameters. The body weight of albino rats were measured up to 14 days, and it is clearly observed that the treated seeds or processed seed extract significantly increase the body weight. Okay. This column is for 14-day body weight, and in organ weight also it has been observed that the processed seeds significantly increase the weight of liver as well as kidney compared to the control one, that green one. Now the blood has been collected from the red groups, and it was the hemoglobin content, RBC content, total blood count, and total blood cell count and platelet count was measured using the standard parameter, and there was no significant difference was found between these 3 groups. the major toxicity effect on two vital organs, liver and renal, that is kidney function, was observed, and the major or the key enzymes of liver and kidney was the amount of this enzyme was measured, and it has been shown that the raw seed extract significantly decrease the amount of SGPT, while the treated or processed seeds decrease the amount of creatinine in seed extract. That means it has some effect on the renal function. And at last, the histopathology study was carried out on the vital organs, that is liver, lung, heart, kidneys, stomach, and spleen, and there was no significant, there was no damage was seen on vital organ. And mucuna is also, mucuna was used by the ancient people for that. Mucuna can increase the reproductive function. So we have also included the epididymis, ovary, and testes to check the toxic effect. And there is no significant changes were observed on the morphology of such organs. And in conclusion, our study observed that the processing effectiveness of the method was defined based on the two most important parameters determining the quality of processed mucuna seeds, and these two are the protein content and L-DOPA content, and other antinutritional components such as phytic, tannin, oxalate, and phenolic compounds. And they were significantly reduced by our treatment, and during the processes, some reduction of crude protein was observed, but in some treatment, this is negligible. And we can use these treated seeds as a human food as well as an animal feed material to cope up with the malnutrition, and we can also improve the food security. Thank you very much.
Thank you, Ms. Shweta.
That, sir, you are not audible. Hello, sir.
One more important thing, please conclude your presentation within 8 minutes and 2 minutes for questions answer. Otherwise, your marks will be deducted. Next participant is Fozia Chaudhary. Next participant, Fozia Chaudhary.
Yes, sir. अनिल इधर आओ जरा. यहाँ से होता है इस शेयरिंग स्टॉप नहीं करना ना. वीडियो को तो ऑफ करो. यहाँ से वीडियो ऑफ करनी. Hello.
Yes, audible. Please proceed.
Thank you, sir. Good afternoon, everyone. Firstly, I thank Dr. Siddharthan for giving me an opportunity to present my research work on this conference. Today, I am here to present a piece of my research work which is entitled Study and Community Diversity of Soil Inhabiting Nematodes of Natural Vegetations of Gulgulabgarh area of Riyasi district, Jammu and Kashmir Union Territory. First, introduction. Nematodes are eel-like, mostly microscopic and ubiquitous organism. They are most successful invertebrate organism after insects and occupy lower position in taxonomic hierarchy. Adapted to and capable of living in almost any possible habitats ranging from deep sea trenches to highest elevations. Being the most abundant metazoans on Earth, nematodes form 4 of every 5 multicellular organisms on this planet, occupy a key position in soil food web, helps in decomposition and mineralization. On the basis of trophic groupings, nematode may be of 5 types, such as bacterivores, plant parasitic, omnivores, predatory, and fungivores. Then, materials and method. Collection of samples. A survey was conducted during September 2021 for enumerating the diversity and community structure of soil nematodes in natural vegetations of Riyasi district. Sampling design was random and 40 soil samples were collected randomly from the natural vegetation area of Gulgulabgarh using trowel. These samples were collected in airtight plastic bags in order to retain the moisture, and the bags were labeled with the names of collection site, temperature at the time of collection, and dates. Within the natural vegetation, samples were collected from the forested area along with the grasslands with Pinus roxburghii, Pinus valenciana, Pinus smathiana, etc. Processing of samples. After the collection of samples, various techniques involve sieving and decantation techniques of cob and modified technique of Biermann was used for extraction and isolation of nematode in the laboratory. The dissected and isolated nematodes from the soil sample were killed and fixed by using the hot formalin acetic acid 4:1 ratio fixative and then dehydrated in glycerin alcohol 5 pints of glycerin plus 95% of 30% ethanol. Identification and isolation of nematodes after fixation and dehydration, permanent slides were made, and then Olympus BX5 and DIC microscope was put in use to study these slides. Nematode counting. Nematode abundance of each sample was counted by preparing slides of nematode in order to identify various genera. Syracuse counting dish was also used to count the nematodes. Community analysis was calculated using following parameters. Frequency, it is the total number of sample in which a particular genus was present. Absolute frequency, it is the frequency of genus upon total number of counted samples into 100. Relative frequency, it is the frequency of particular genus upon total frequency of all genera into 100. Mean density, it is the number of nematode specimens of a particular genus counted in all samples divided by total number of samples counted. Relative density, it is the mean density of a particular genus, sum of mean density divided by sum of mean density of all nematode genera into 100. Tomer et al. 2006. Diversity indices, following diversity indices were calculated: Shannon-Wiener index and Hill index. Then results, diversity of nematode genera. In this nematode community diversity study of natural vegetation of Gulgulabgarh area of Riyasi district, we obtained 30 nematode genera with bacterivores representing the highest number 57.8%, followed by omnivores 22.23%, predators 11.95%, parasitic 5.8%, and fungivores 2.85%. In terms of taxonomic grouping, the order Reptilidae representing 44% of the all collected specimens, followed by Dorylamida, Tylenchida, Amonentrida 10%, and Aphentrida 3.3%. On the basis of trophic grouping, bacterivores represent 50% of the collected nematode, followed by omnivores, parasites, predators, and fungivores. Among the bacterivores, Cuticularia was the most dominant, followed by Mesorhabditis. Discussion and conclusion. Soil ecosystem inhabit and support a rich diversity of fungi, bacteria, algae, protozoa, arthropods, mites, nematodes, etc. Nematodes contribute directly or indirectly to the biogeochemistry of the soil by regulating decomposition and nutrient cycling. Their diversity and abundance depends upon ecological and edaphic factors like soil pH, nutrient content, moisture, etc. The other factors which influence their richness and diversity are soil type, pore spaces, root secretions, plowing, tilling, anthropogenic disturbance, grazing, pesticides, etc. In the present study, the number of genera identified was 30, representing bacterivores nematode 15 genera, omnivores nematode 5 genera, Herbivores nematode 4 genera, predating nematode 4 genera, and fungivores nematode 2 genera, which were the least abundant. Among these, bacterivores represent the highest number of genera. Since the soil was found to be mildly acidic, it supported the bacterial growth, and as a result, bacterivore nematode represent the highest number. Again, fungivores nematode were least abundant because they thrive well in more acidic soils. In conclusion, our study on natural vegetation area of Gulabgarh revealed rich diversity of reptided nematode and lower diversity of monantid nematodes. Apparently, this could be attributed to anthropogenic disturbance, among which tourism might play a key role. As far as the nematode diversity is concerned, the present study can be considered as an initial step to study this area. Further research on this area is warranted in order to fully explore the biodiversity of this region. Thank you.
I have asked one question. How
Yes.
your work is used in daily life or in normal routine?
जी
What
sir.
is your work
जी जी sir.
is used in daily life or routine life or normal human mankind's?
Sir, actually, these are the nematodes are used as a biological indicators, soil health indicator, which types of soil is there. Several studies on forest diversity of nematode, and our study revealed that the soil was rich in organic humus, which said that the bacterivores nematode were the most abundant and fungivores were the least abundant.
Is this type of indicators are not available in commercial level?
Commercial level.
Or
Sorry, sir.
same type of product? Is your product is novel?
Yes, sir.
Is your product is novel? Is same types of product are not available in market?
No, sir. They are living things, sir. Actually, these are the microscopic organisms that reveals about the soil conditions, soil moisture content, which soil type is this, acidic soil, सर different bacterivores जैसे सर कुछ हैं जो हमें सर omnivores वाले हैं, वो कहाँ पे मिलते, कौन सी soil में है, तो उनको सर हम biological indicator ऐसे use कर सकते हैं सर nematodes को.
Okay, thank you so much.
Thank you, sir.
Next participant is Miss Chandana HM.
Good afternoon, everyone. Myself Chandana, and I'm here to present my seminar on study of fish parasite, which include both ecto and endoparasite. This is my abstract. Coming to introduction, fish constitutes an essential part of human diet because due to its easy digestibility and it has high nutritional value. It is important because it has high quality of protein for humans, especially cultured carbs are susceptible to various kinds of disease such as bacteria, viral and parasitic diseases. First of all, parasite is an organism that lives in a host organism and derives its nourishment from the host and gets benefit from the host. Parasites are divided into ecto and endoparasites. Ectoparasites are those parasites which live outside of the host. For example, fish lice, Trichodina indica, and crustacean parasites like Caligus crucino. Endoparasites are those parasites which lives in the internal organs like digestive tract, body cavities, and other tissues. For example, tapeworm, roundworm, and acanthocephalans. These parasites affect the growth and survival of the host and reproduction, and it also affects the marketability of aquaculture and fisheries products. The main objective of this review is to know the parasites of fish, which include both ecto and endoparasites. Literature review. Fish have a full range of disease like all animals, and many of these are due to external agents like bacteria, virus, and fungi, and other arises internally. The fish which is affected by the parasite shows symptoms like weight loss and abnormal behavior, and it eventually causes an economic loss in fish farming sector. Parasites are divided into two distinct groups, namely ectoparasites and endoparasites, according to the physical characteristics of the parasite and their life cycle and host infection site. Ectoparasites are located on the external surface of host organism. It is one of the most hazardous threats to fish health. They have been noted by many researchers as one of the major problems in pond fish culture, where high temperature and organic content may accelerate the life cycle of these parasites and promote their spread. Uh, some of these uh, some of the examples of ectoparasites are Trichodina indica, Ichthyopteris multifilus, Chilodenella cyprinid, Orgillus species, and crustacean parasites like Caligus crucino, Bomolochus decapteri, Lernanthropus illice, and Pisicola geometra. Some of these parasites are attached to exposed body surface, or they live in gill chambers and feed on body mucus and blood. Fish lice. It is one of the ectoparasites. It is commonly called as fish lice. It is dorsoventrally flattened. It consists of 8 legs for swimming purpose. 2 large sucker is present. Hargulus has direct life cycle because it does not require any intermediate host. Because of their size, Hargulus can be diagnosed with the naked eye. Fish leech Pisicola geometra. It is worm-like and has large sucking disc at each end. It grows up to 20 millimeter and feed on the blood of its host. Anchor worm, Lernae. Lernae species are commonly known as anchor worm. It is one of the crustacean parasite. It can grow up to 12 millimeter. Whitish green thread-like structure hang out of the fish skin with an inflamed area or ulcer at the point of their attachment. Endoparasites are located in the body cavity, like body cavity, intestines, and various internal organs, and sometimes it can be seen in flesh. Some of the examples of endoparasites are nematodes, cestodes, and acanthocephalans. The helminth parasites that infect two vertebrates, which belong to two phyla, Platyhelminthes and Aschelminthes. In general, the endoparasitic helminthes have heterogeneous life cycle. Heterogeneous life cycle means it requires at least two kinds of host to complete its life cycle. Trematodes, it comprises of monogeneans and digeneans. Monogeneans trematode are also referred as flukes. They commonly reside in skin, gills, and fins of the fish. Digeneans, which means they have essential tegument with two suckers. One is present at ventral and one is in oral. Monogeneans have direct life cycle, hence they do not require any intermediate host. Cestodes, they are also called as tapeworm, and they are ribbon-like flatworm. They are especially infect alimentary tract, muscle, or other internal organs of the fish. They require one intermediate host to complete its life cycle as adult in the definitive host. Nematodes. This phylum Nematoda is one of the most common phyla of animals. It contains both free-living organisms and parasites of plants and animals, including fish. They are elongated and cylindrical in shape and unsegmented, bilaterally symmetrical with a complete digestive system. Acanthocephalus. It was first recorded by Coelrettu from the intestine of the fish as a parasite and proposed the name Acanthocephalus in 1771. These are also known as spiny or thorny-headed worm. Their body is divisible into trunk and retractile proboscis. All are intestinal parasites. These are cylindrical worms with anterior part which consists of an eversible hooked proboscis. They absorb the nutrient materials from their body surface. I conclude my topic. Parasites that affect the fish includes parasitic protozoans, acanthophilins, nematodes, digenins, cestodes, and crustaceans, which are the important parasites of fish. Helminths are highly specialized parasites that require specific definitive host. These parasites causes diseases such as trematodiasis, cystodiasis, and nematodiasis. These diseases are mainly through eating or raw or undercooked fish. Therefore, awareness should be raised about fish parasites and consumers should not eat raw or undercooked fish. These are my few references. Thank you.
Hello. Sir, can I ask some questions? Hello. Okay. Sir, can I ask some question?
Yes, ma'am. Yes, ma'am. Please. Yes, ma'am. Please.
Sir, my question is that can you give me example of nematoda, cystoda, acanthophilins? Can she give me examples? One of example.
Example for cestode is tapeworm, ma'am.
Okay.
Nematode roundworm.
Yes, I know they are commonly known as tapeworm, roundworm, but you didn't give any particular example of parasitic species of those. It's okay now. Okay, thank you.
Thank you, ma'am. Our next participant is Sareen Fatima Rijvi. Sareen Fatima Rijvi.
Yes, sir, I'm present.
Yes, please proceed your presentation.
Sir?
Yes, yes. Yes, please share. You are audible.
Is my presentation visible to you all?
No, 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 not visible.
Okay. Can you see my poster?
Yes, please.
Not visible. Okay. A very good afternoon to one and all. Myself, Sharing Fatma Rizvi. I'm a research scholar from Integral University. I'll be presenting a poster on my research, which is valorization of chicken feather hydrolysate as a substrate of PHAs.
Ma'am, your
PHAs
poster is
are
not
polymers
visible.
which are gained. My poster isn't visible. Wait a minute. But I'm sharing the whole screen. Share. Is it visible now?
No, no.
I don't know what is wrong.
Please send me your poster at organizer भावना और करिश्मा.
Okay, I'll do that.
Next participant is Amina. Amina Sahib. Next participant Amina Sahib. Are you there? Are you hearing me? Next participant Rohit Goet.
Hello.
Next participant Rohit Goet. If you are ready, then please proceed.
Okay, sir, I am ready.
Yes, please proceed.
Sir, is my PPT visible?
No, not visible. Yes, visible very much.
Good afternoon to all of you. I am research scholar under the guidance of Dr. Joginder Singh, Department of Chemistry, Mercy Markandeswar, deemed to be University Maulana, Ambala. Today I am going to present, my title is Development of Polyether sulfone based hybrid membranes using graphene oxide, zinc oxide, nanocomposites for enhanced anti-fouling control of the membranes. First of all, contents. We will discuss about the introduction section, techniques of graphene oxide synthesis and graphene oxide zinc oxide nanoparticle synthesis, characterization of the compounds and the fabricated membrane, and conclusion. First of all, introduction. The presence of different type of toxic and harmful materials such as dyes, heavy metals, and other colored materials or organic compounds in the water bodies are very harmful for the human as well as other ecosystem around us. They are mainly found in wastewater influents, which was come from the industrial waste, paint manufacturing industries, paper and textiles, dyeings, and cloth dyeings industries.
Please
Too
change
much
your slide.
exposure. Yes, sir.
Please change your slide.
Yes, sir. Sir, I am discussing on the first slide, introduction
Okay.
section. Too much exposure of dyes and heavy metals can cause many diseases such as jaundice, nausea, dead tissue, increase our heart rates, and other noticeable effects on human health. Some heavy metals like copper and nickel are carcinogenic for human health. So we have a very huge need to contaminate the fresh water, and we need fresh water for our daily needs. So we need to fresh it from contaminated waters, and many technologies are developed for treating the contaminated water. Some common techniques are biological, physical, and chemical processes such as chemical oxidation, coagulation, separations, microbial degradation, photodegradation, and membrane separations. Membran separation is one of the best techniques due to its cost-effective nature and its purification and removal efficiency of dyes and heavy metals. And the membrane is very easy to prepare and very easy to use. Membrane is commonly used in RO for nanofiltration and ultrafiltration technologies. In comparison to other techniques such as powders, particles, gels, polymeric membrane are more widely adopted in water treatment due to their porous structures and good filtrations. First of all, we synthesize graphene oxide. For the synthesis of graphene oxide, we use Hammer's method. In the Hammer's method, we use graphite powder plus sodium nitrate plus sulfuric acid and potassium permanganate. By this method, we get the synthesis of graphene oxide. It is very safe and time efficient method, so we prefer this method. Mainly graphene is synthesized by several methods, but hammer method is the best method in the way of time and safety. Then we use the synthesized graphene oxide for the synthesis of graphene oxide and zinc oxide nanoparticles. First of all, we take 200 mg of synthesized graphene oxide powder, which were dispersed in 20 ml distilled water at room temperature and for 40 minutes. Then the obtained suspension was transferred into airtight glass bottle and stirred for 2500 rpm for 15 minutes at 30 degrees Celsius for proper exfoliation of graphene oxide. Then we add zinc chloride and 1.0 gram NaOH sodium hydroxide pellets in the suspension. The resulting mixture
Sir, your
was sealed.
slide is not visible.
Sir, I
Only
am
first
sharing my slides.
Only first slides are visible.
Is it not visible, sir?
No, not visible.
Sir, I am sharing my slides and changing my slides.
Yes, now visible.
After that, the resulting mixture was sealed in air glass tight bottle and stirred for 7 hours at 65 degrees Celsius, then allowed to make cool at room temperature. The obtained product was filtered with Woutman filter paper and washed with distilled water and pure ethanol 4 to 5 times and dried in the muffle furnace at 450 degrees Celsius to get the graphene oxide, zinc oxide nanoparticles. After that, we use the nanoparticles in the fabrication of membrane. For the fabrication of membrane, phase inversion method was opted, and first of all we use polyether sulfone pellets, which were dehydrated at 80 degrees Celsius in the oven for 2 hours, then dissolved in the solvent DMF and for 8 hours at 60 degrees Celsius temperature. Then we add PVP and graphene oxide zinc oxide nanoparticles 1 gram in the solution and further stirred for 2 hours. Then we fabricate the membrane with the help of phase inversion method. Then the characterization of the nanoparticles and the fabricated membrane. First of all, we go for the nanoparticles and we characterize the nanoparticles with the help of FTIR, FESAM and EDX. From the FTIR, we see that a broad peak of OH or hydroxyl group at 3400 to 3000 range in the FTIR spectra, and the graphene oxide and ZNO below 1000 at approx 450 nanometer inverse. The prepared membrane was also characterized by the hydrophilicity, pore size, mechanical strength, and water permeability of the membrane. This is the EDX and SAM images of the nanoparticles. From the EDX, we can see that there is carbon, oxygen, and the zinc compound in the nanoparticles. And petal-like morphology over the graphene oxide sheets confirms the graphene oxide zinc oxide nanoparticles formation. After that, we characterize the membrane with the help of FESAM images. From the first images, we can see that the pure polyether sulfone images have very rough surface and very small pore size, while we use graphene oxide, zinc oxide incorporated in the membrane of polyether sulfone, and after the incorporation of graphene oxide, zinc oxide in the membrane, the pore size of the membrane was increased, the surface becomes smoother, and in the cross-sectional image, we see the finger-like projections or thick top layer as comparison to pure polyether sulfone membranes. Further, we use these membranes in the filtration system, and in the filtration system, we use these membranes for the enhanced removal of dyes and nano heavy metals. From the filtration system, it was concluded that about 97% of methylene blue and 86% of copper divalent and nickel divalent ions were removed with the help of graphene oxide, zinc oxide nano filtration membrane. In the last slide, we go to the conclusion. In the conclusion, the effect of graphene oxide, zinc oxide loaded nanocomposite materials for the performance of polyether sulfone, zinc oxide, graphene oxide was investigated in this study. The best performance membrane was dedicated to polyethersulfone membrane blended with zinc oxide nanoparticles decorated on graphene oxide. The increasing the zinc oxide decorated graphene oxide nanocomposite material up to 100 mg led to a maximum in membrane permeability 5 to 0.35 liter meter square per hour as compared to pure polyether sulfate membrane. Thank you.
I have asked some questions. Actually, I have some
Yes, sir.
doubts.
Yes, sir.
Why you used hammer method, not used modified hammer method or advanced hammer method?
Sir, actually there are three methods in the literature: Stonemeyer method, Hammer method, Brody method, or modified Hammer method, Tours method. We use Tours method or modified Hammer's method, sir. Hammer's method will become in 1958, or improved Hammer methods will become in 2010. We use 2010 Hammer's method, which is
But
very.
after that 2015 or 16 advanced hammer method are available.
Sir, we are not using the advanced method. I don't know about that. We used 2010's Marcano and Tours Hammer method for the synthesis of our graphene oxide.
Okay, and graphite powder you are commercialized or making
No, sir,
from some biochar?
graphene powder we take from the Sigma Aldrich, and graphene oxide we prepare from each. And we also publish a paper in the environmental research from the graphene and its antibacterial activities and its properties for the uptaking of pesticides.
And last one more doubts. Your product are absorb 80%, 86% methylene blue.
Ji, sir.
And normal graphene oxide and graphite absorbed 78 to 79% methylene blue. Then how your product is novel or used?
Sir, actually we also characterize the membrane with the zeta potential, and the zeta potential of our membrane is minus 21.9 megavolt, and we can conclude it from there is pi stacking. The aromatic rings are present in the graphene oxide, and the aromatic rings are also present in the dyes formula. There are also the electrostatic interactions. These are the interactions which enhance the removal efficiency of dyes from the water due to the graphene oxide and zinc oxide nanoparticles.
Okay.
In the membrane.
You do not have add a characterization of graphene oxide?
Preparation of graphene oxide, sir, it is in the very short form because we already published 1 or 2 chapters on that work. We are further working on graphene oxide hybrid membranes further.
Okay. Thank you so much.
Okay, sir. Thank you very much.
Next participant is Amina Sahib. Next participant, Amina Sahib.
Yes, sir, I am here. Good afternoon,
Charisma ma'am, please share the PPT. Charisma ma'am, please share the PPT. Charisma ma'am, please share the PPT. Okay, please contact her. Next participant, Sarin Fatima Rijvi. Sarin Fatima Rijvi. Your screen is not visible.
Is it now?
No, not visible.
I don't know what is the issue. Is it now visible?
No, not visible.
Okay, I'll contact Bhavna.
Next participant, Anil Bhardwaj. Next participant, Anil Bhardwaj. Yes, sir, please proceed. Sarin Fatima Rajvi and Amina Saheb, please contact Bhavna. Please unmute yourself.
Sir, am I audible?
Yes, yes, very much audible. Please proceed.
So, thank you so much, sir, for organizing such a wonderful conference. Myself, Anil Bhardwaj, and my present work is on the comparative study of soil nematodes of upper and lower regions of district Udhampur, J&K. So basically, we worked on soil nematodes. Soil nematodes belong to phylum Nematoda from Greek nema, a thread, a long, thin, often thread-like worms. They are microbial feeders. Some are parasitic, some are free-living, and they are basically microscopic and eel-like roundworms. So, introduction: the group of roundworms represents a major component of the below-ground fauna diversity. Nematodes include more than 27,000 described species, are found almost in every inhabitable place on Earth, and represent about 80% of below-ground bulk metazoan taxonomic and functional diversity. Nematodes on Earth, most abundant metazoan and universal in soil environment. Their size is large enough to sufficiently seen by light microscopy to small enough which can be sufficiently inhabited by water films surrounding soil particles. So my study area was basically the Shipalik range, a group of mountains in the Lesser Himalayan region in Indian territory of Jammu and Kashmir. So this study was conducted with average elevation varies from 682 meter above from sea level to 2592 meter. Soil samples were collected from 6 different regions: Udhampur, Ramnagar, Majalta, Basantgarh, Panchari, and Chinani. And so nematodes were sampled across 6 different locations. From both upper and lower regions, 50 samples were collected. In subalpine areas, soil samples were predominantly collected with Ficus, Pinus, and the lower regions were mostly dominated by the agricultural fields. So after the soil sampling, we use fresh soil. Soil was used for extracting soil nematodes using the sieving and Bierman funnel method. All the nematodes in each sample were then counted under a Olympus stereosome SZ16 microscope mounted into slides for identification to the genus level and assigned to various functional grades based on the trophic group and life history strategies. Another sub was used for measuring soil parameters including soil humidity, pH, conductivity, and temperature. So nematodes communities functional characterization. Depending upon the abundance of functional grades of nematodes, phase indices were calculated so as to analyze the functional role of nematode based on food webs along various mountain transects. All the identified nematodes were classified into five main trophic habits. They are bacterial feeders, plant feeders, omnivores, predators, and along the colonizer persister scale. So basically, this is the CP scale. Two major indices of relating nematodes functional groups to ecosystem functioning: the sigma maturity index and enrichment index. So the sigma maturity index representing the proportion of the different CP groups for the whole nematode community, where higher values indicate the nematodes harboring persistent life history traits are predominant with each of those different nematodes categories. The enrichment index, which is based on the biomass of opportunistic nematodes that respond rapidly to the increase in bacterial and fungal population that arise from the organic matter decomposition. High value indicate high soil enrichment and high fertility. Biomass values were extrapolated from Ninja software. This is the location from where the samples were collected. These 6 different sites. In terms of diversity, the percentage of soil nematodes from the agricultural field, where the Rhaptidida roundworms basically they were the dominant, and the Rhaptidida was followed by Dorylamids, and then the talent Mononcida was the least dominant. So this is the chart, diversity percentage chart, and in this the forest, basically the nematodes. 41%, basically the bacteriovores were the most dominant in the forest soil, and the fungivores were the least dominant. So the trophic groups of these nematodes from the lower regions. In the agricultural field, the Rhaptidid roundworms were the most dominant. Basically, they are the bacteriovores, and the predatory, like mononcs are the predators, and they were the least dominant in the agricultural field. So trophic diversity of the upper regions. In forest also the bacteriovores were the most dominant, and followed by plantic. Predatory also were dominant, but the fungivores were the least dominant in the forest soil. So the result, the following were the parameters were used into taken into consideration: soil climate covariation along elevation. We found that the elevation was the correlated with soil moisture, pH, temperature. The temperature decreased in average by 12.5 degrees. Conductivity also decreased. pH also decreased as we move from the lower plains to higher plains, and the moisture is also increased along the elevation gradient. As in the upper hills, due to continuous rainfall, the moisture will increase. So we observed a general increase in the number of nematodes along the elevation gradient, but the diversity of nematode communities declined at the highest elevation, which mostly consisted of herbivores and omnivore genera. Among soil variables, pH was negatively associated with fungivores, but positively associated with herbivores, while conductivity and temperature were positively associated with bacteriovores. And finally, soil moisture was positively associated with omnivores. We found that the indices related to ecosystem properties varied with elevation, with nematode communities being associated with more productive and mature ecosystems found more likely living at low versus high elevation. Especially the maturity index showed an increase along the elevation gradient, depicted an increase in the relative abundance of persistent nematodes when moving up along the elevation gradient. So these were the diagrams of the charts which show the CP scale values and other the R and K strategies nematodes. Basically, the K strategies basically belong to the upper region and they are more stable as compared to the agriculture, then where the soil is more disturbed. So where the decomposition is more and the organic matter is also more. So which favors actually the bacterial growth. So in conclusion, we have showed that along elevation gradients, soil nematodes are particularly good by indicators of local ecosystem properties. Particularly, we show that the ecosystem along high elevation, such as wider range of functional and taxonomic diversity than their respective low elevation sites. These results are in line with previous findings obtained along elevation gradient in across broad latitudinal gradients, and future work should focus on extending similar research to study the stability of such patterns across sites and years. Indeed, mountain ranges are unusually biodiverse, which copies accumulation of the endemic species, which is the reflection of high variation in hydrology, in meteorology, as well as in ecological and evolutionary process. Accordingly, mountains plays an important role in sustaining Earth's biodiversity and ecosystem functioning. Soil nematodes contribute tremendously to such diversity in both terms of both taxonomic and functional diversity. Therefore, a better understanding of the causes that generate nematodes biodiversity can inform on impact of climate change and land use change on ecosystem functioning worldwide. Thank you.
Thank you, sir.
Welcome, sir.
Bhavita ma'am asked any questions?
Sir, what is the
Hello.
process of extraction of nematode from the root of the plant? Or
Basically,
those parasites from which part of the plant?
basically rhizomes, जो होते हैं.
But you didn't mention anywhere rhizome. Somes, is that the right word? Or it is rhizosphere?
Rhizosphere, sorry ma'am, yeah, you're right ma'am, rhizosphere.
Okay. And from which part of the plant you extract these parasites? Can you tell me that?
Ma'am, your voice is not audible.
Can you please tell me from which part of the plant these nematode are extracted out?
Okay. Hello.
My question is that from which part of the...
Okay, ma'am, I understood your question, but my work, I work on replicated roundworms, and they're free living. So we haven't done any extraction of plant parasitic nematodes. So basically, hello.
Anji, please carry on.
Yes, ma'am. So they basically, I think, attacks the rhizosphere part of the plant roots. So,
okay.
Yes, ma'am.
So they are extracted from the soil.
Work is also on replicated roundworms. So and they are bacteriovores, free living nematodes.
They attached with the roots.
Hello?
Is that? They are free living and they are attached with the root. Is that true?
No, ma'am. Plant parasitic or not free living, they are parasitic nematodes. But my work is on free living nematodes, basically replicated roundworms.
Okay. These are these are also present on another part of the plant. My question is that is you recorded them from any other part of the plant or they are typically found in the roots?
No, ma'am. We have collected soil samples.
Okay,
and,
you collected
not specifically from the roots of the any plant, but in general we have collected the soil samples from different locations, like I said, that from agriculture field and from the forest soil. So in actually the forest, we have seen and we have collected, we have seen plant parasitic also in our research, but they are mostly like from forest area basically. In organic rich, where the organic matter is more, which actually favors the decomposition, where we have found the mostly the bacterioborne nematodes.
Sir, can you give me please any one of the example you got from those area soil?
And plant parasitics.
Soil nematode which you extracted from the soil of that area.
Yes, ma'am. Like rhabdated, we have seen Uticularia, Diploscepter, Mesorhabditis, Rhabditilla, Wilsonema, and Bunonematoids. And plant parasitic like Helicotylecans. In general, we have seen a robust population of Helicotylecans.
Plant parasitic. Plant nematodes. Okay. ठीक है. Okay, thank you, sir.
Okay, ma'am.
Thank you.
Okay, welcome, ma'am.
Thank you. Next participant, Dr. Nikul Chawada from Gujarat.
Am I audible, sir?
Yes, sir. Very much audible.
Okay. So, sir, can I? Okay. Thank you, sir.
Yes, please.
Okay. Sir, I try to share my PowerPoint presentation.
Okay, please.
It is visible, sir? Hello.
Yes, sir.
Okay. It is visible, sir. Am I right?
Yes, visible. Please proceed.
Okay. Thank you, sir. Thank you. So requested Siddharth sir and their team, faculty members and students, also research scholar. So good afternoon to everyone. My research project based on the diversity of phytoplankton and how the phytoplanktons, and right now we are aware about the planktons because they are primary food producers in the water levels. So they are very much important chain of food chain, and it's a very important and living part of our research. Actually, sir, this is a research project which is given by the government of Gujarat. And right now we are just working on the physicochemical properties, how the physicochemical characteristic like abiotic factors, which influence the biotic factors. In the biotic factors, we are studied on a phytoplankton diversity, on a zooplankton diversity, and also the microbial biodiversity. Because each and every diversity, they are correlated with each other. Sometimes what happens, if I am talking about the Triveni Sangam river, and actually it is a very located, it's a Triveni Sangam, it is a estuaries, and it is very near to the Somnath temple, and it is a holy place. So it's a holy place, and many of the worship is going on by the Hindu communities, and due to them, they are just putting some gulals and abils they are in the waters, and due to them, some influence, some biodiversity will fluctuate because those all are the chemicals, and those chemicals which are directly or indirectly influence the biodiversity of the planktons. Okay, so basically this project is based on how the abiotic factors playing an important roles. So we are taking the 15 parameters, that is abiotic factors which are included pH, temperatures, and many others. And we have studied their influence in phytoplanktons diversities. So here my three students, and I'm also doing this research work. Still, this process is going on. My remaining three members, they are right now they are doing the exam, so they are not attending this part. But as apart from them, I'm representing this work. Okay. Now, the second thing. Yes. So these are my contents, that how we are collecting the samples and introductions of my research works, methods and methods, the result and conclusions, and many others. Now it is the abstract. Now you can see that the Triveni Sangam connected the three rivers, that is the Hiran, Kapila, and Saraswati. These three rivers, these are they are confluence on the west coast of the India. This is where they meet the Arabian Sea and form an estuaries. And the plankton, they are very important role, because I'm already told that in the slide that the planktons, which are the playing a very important role, they are primary food producers and they maintain the total whole ecosystem. So the planktons, what are the planktons? And basically our work are on the phytoplanktons. Okay, so we are isolated, or we are identified the 35 phytoplankton species we are identified during the analysis, including the Rhizocelinus species, these species are there, and Melis also species are there. So we are observed that during they are dominant. We are selecting the 3 different locations and collected the water samples and studied their physicochemical properties, and also we have studied their phytoplankton diversity. Can you imagine, sir, where this worship process is going on? जहाँ पे मतलब जो हल्दी कुमकुम और पूजा, जहाँ पे पूजा पाठ वहाँ पे होता था, where they are the peoples who are spreading such a waste in particular point. We are find out very less types of phytoplankton diversity. So can you think about that, how they are influenced? Okay, by the physicochemical parameters, or the physicochemical parameters of the water is changed, then how they are influenced. Also, we are doing the bacterial analysis, bacterial concentration of different types of the bacterial type. We are also isolated from that, but right now I'm not discussing about that. Okay, now the introductions, that is, we know about the phytoplanktons. There are phytoplanktons, that is zooplanktons and phytoplanktons. And in the phytoplanktons, we are including the diatoms and diflagellates. Okay, so in present study, we examine the relationship between the abiotic factors and phytoplankton diversities, because directly or indirectly, definitely they will interact with each other. And planktons are very important. Why we are choosing the planktons? Because they are very important. They are primary food producers. They maintain the several ecosystem inside the water bodies. Okay, so now the supplying the significance, we are the studying of the freshwater rivers, supplying the significance to the suctions and the dynamic of the zooplanktons and the planktons. They are key producers of an aquatic ecosystem that organize a biological productivity. So these are the metals method, how we are collecting the water samples. Here I am just GPS location is given at here, because 3 locations we have selected, 3 location points we are collected the water samples. We are divided the water samples in a 2 part. The first part, the 5 liters of the water for physicochemical analysis, and another 1 liter, or more than 100 liters, we are passed out from the plankton net and collected the 250 ml of the water. They're performing the drop count method to analysis the which kind of the planktons which are present in the water samples. The physicochemicals analysis of the water samples. Here we are just studying the pH, conductivities, and estimate of the TS, TDS, chlorine concentrations, water hardness, estimation of the DO and DODs. So all this process, all the physicochemical parameters we are done by as per according to the output. Now the second point is the plankton analysis. We are performing the drop count method, which is very much important, and we have the plankton manuals to identify all such a kind of the species of plankton. Now, yes. Now, what is the conclusion of our this methods, and how we conclude that how this abiotic influence, abiotic parameters playing an important role in maintaining the ecosystem or the diversity of the planktons. So, sir, actually here we are just conducting this research pre, middle, and post 2021 to 2000 here. Sorry, by mistake, it is 2022 in during the time of the winter. Right now, after the winter, this process is going on. Same this process is going on the summer. Same this process is doing during the monsoon season. So I'm already told that the Hiran, Kapil, and Saraswati river, where the water is mixed and they are also mixed with the seawater, an Asheris point is formed. We collected the soil samples and 15 parameters about parameters we have conducted. So in the conclusions, we noted that the pH of this water research related to the 7.8. The pH is the temperature is around 21 to 25 degree temperatures. So it is a normal winter temperature in Gujarat. So pH of this wetland water is 7.2 to 8.9. Here the 8.9 we are noted this during the January of the water sampling time. So 8.9 pH is indicated that some higher salt concentration which are present in this water. Same the conductivities also. Here I'm just write down that. Yes. Yes, you can see that here I'm just putting one table that the pH range is there. Then the conductivity is also we are measuring in this the micrometers and the centimeter. Then the TDS level and the DO, then BOD. The issue is there before the 3 to 5 years when I was conducted this study, the DO concentration is very good. But after this after 2 to 3 years, then I noted down that the pH sum of the level there is a location 1, 2, and 3 above the date is given. Okay, so you can easily understand how this all the physical chemical parameters are mentioned. Here, the DO at the biological oxygen demand data indicate that the dissolved oxygen level, which is range is 1.96 to 7.92 microgram per liters. Higher DO value indicates the good conditions of the aquatic life. We already know that. Then the TS and TDS, where the TDS of the water samples, that is the range was measured 1110 milligram per liters to range of 1325. Okay, so the TS and TDS is higher as compared to the normal water because it is the estuaries and water. So this water is cannot directly used for the agriculture purpose, and also it's a drinking purpose. Now the water hardness, second parameters, that is related to 3000 to 10,000 milligram per liter. So the higher salt concentration is already measured. Same due to the conductivity is higher, the salt concentration is higher. We are also studies of the chlorinities and salinities and alkalinity, acidities, and NaCl concentrations we are noted at here. So these are the parameters. Then and already if I'm taking the single parameters related to pH and dissolved concentration of oxygen, I am find out the more concentrations of the dissolved oxygens where the diversity is good. Okay, here the 7.92 milligram per liters, where the concentrations of phytoplankton species are more compared to the other. Okay, but these are the samples point where we are conducting this research in this field. Now, after this, yes, the diversity. Yeah. Yes, conclusions. So the limnology analysis of the Triveni water result indicated that the physicochemical parameters can influence the biodiversity of the planktons. I'm already told where the worship process is going on, where this process is going on. We observe that the plankton diversity is less. And what happened when the phytoplankton is less? Comparatively, zooplankton is less. If the zooplankton is less, comparatively fish is there because each and every are the very important ecosystem. They are connected with each other. So it is the very important part of that. And the diversity of the plankton directly or indirectly increase the diversity of the zooplankton and also impact on feed diversity. Bacteria, we are identified the more than 8 species of the microorganisms. Sometimes we are also identify the cholera, that is Vibrio cholerae, we are also isolated from there. And Staphylococcus aureus also we are isolated from that water samples. So it's indicated that this water is polluted microbial biologically, and plankton diversity is less in such a point where this activity is going on. So these are the 4 species where we are getting the higher concentration during our analysis. And so this it's nothing but conclusions, and I did discuss about that conclusion. And these are the physicochemical parameters and how we are different 15 types of the parameters we are studied. And second is the plankton diversity. You can see that different types of the groups I'm just giving. That means how much amount of the different species are we are isolated. But we are isolated of the rhizoselenia group higher concentration, but sometimes what happen, it can also cause algal bloom, and due to them they're toxic, they are also release the toxic in the water. That may be issues in they are also affected the diversity of others. Okay, these are my reference. Okay, so thank you very much, and if you have any question then you can ask me. Thank you. Hello. Sir, actually, I hello. Sir, mic is mute, sir. So I cannot hear any question.
मैं उधर जा नहीं सकती, मुझे उधर थोड़ी नेगेटिविटी है।
Amina Sahib, are you there? Miss Amina Sahib. Sarin
Yes, sir,
Fatima.
I'm here.
Yes,
I'm
yes,
here.
please proceed. Please proceed.
Yes. Ma'am, please
Charisma
share
ma'am, please
my
share the slides. Charisma ma'am, please share the slides. Participants should complete your presentation within 8 minutes.
Good afternoon, everyone. My name is Amna from Integral University. Today, I am here to present, give some idea or share my views on biofuel production of by converging of banana peel waste with the help of microorganisms, which is a sustainable approach. Why I focused on a banana peel? Because India is the world largest banana producer. By focusing on its lignocellulose biomass on biofuel production obtained, which is obtained from it. Banana peel waste provide an alternative substrate for ethanol synthesis. Bioethanol production from banana trash is a viable approach to utilize the surplus biomass, which is especially in Indian southern areas. Banana peel is whereas dumped in a very surplus way in waterways and uncontrolled dumping areas, and it is very cost effective also, which can also help in a very sustainable approaches on controlling our pollution also. So first we focus on bioethanol. What is bioethanol or biofuel and why we need it? Bioethanol is a sort of renewable energy resource which may be from microorganisms, plants and animals. It can be obtained from any of the plants which contain a starch like corn, sugarcane, banana peel waste, and having a lignocellulose biomass. It can also synthesize in a synthetic way by a chemical process of reacting ethylene with a steam. So I want to conclude what is the advantages of bioethanol. The invention of bioethanol produce a less reactive and reduce a low amount of the greenhouse gases which is in comparative of burning of gasoline, which is result in a potential of forming of ozone. The use of bioethanol reduce the carbon dioxide emission, which is a most potential renewable energy resource which is mitigate in the energy crisis. Countries that do not have a crude oil and have a less not having a financial stability, then they also grow a crop and use some economical freedoms by using a crop in a eating way, feeding themselves, and the waste product they can use in making biofuels. Now I tell you why I have selected the banana peel as my research approach, because it contains an essential amount of protein and carbohydrates and extremely low amount of anti-nutrients like phytate, alcohols, and phenols like other fruits that act bacterial growth inhibitor, which help in a fermentation process. Now I also tell you that there are it also help in causing soil pollution. So by using this approach we can control on that approach also, and also in environmental cleanness, because the invention or the emission of by burning a biofuel, it can improve the air quality in comparison of burning of gasoline. These benefits make the banana more appropriate over other types of fruit waste, sustainable as substrate in bacterial fermentation for bioethanol production. By literature review, I get a view that 31.4 grams of banana peel waste gives a 15 milligrams of bioethanol production. By use What is the my approaches that first of all to collect the sample, uh, give them a pre-treatment by clearing all the debris and unwanted material having in that agricultural waste, and process is with the hydrolysis, which is a chemical interaction of chemicals with water leading with decomposition of both as well as substance and water. Then it goes through the fermentation process. Then after fermentation it goes through the distillation, in which it converts the liquid into a vapor form. Then after the condensation it also get converted into a liquid form, and the at result bioethanol is obtained. And uh, we conclude that we conclude that by using this approach we can predict that 0.04 gram or 0.21 gram of banana peel gives the approx 1 gram of um, ethanol production, which is found it is sufficient to support the target of ethanol production in India. This study reveals that it is a very cost-effective and abundant substrate, especially from the tropical countries like India. It also helps in economically balanced, environmental friendly, or we can say that it can help in up, obtaining a sustainable approach. Thank you.
Thank you, ma'am. Next participant, Serene Fatima Rejvi. Serene Fatima Rejvi. Next participant, Umi Salma. Next participant, Umi Salma.
Hello, sir.
Yes, please proceed.
Sure, sir.
Sir, it is visible.
Yes, visible, very much visible.
Very good afternoon, one and all present here. First of all, I thank Siddhartha Din sir who gave me an opportunity to present here. I'm Umay Salma and I'm going to present seminar on my project topic study on parasites in the food fishes available in the local markets of the Mysore city. So the abstract includes all our project work research in a very short summary. Then coming on to the introduction part. So our current study was carried out to evaluate the prevalence of the parasites in the freshwater fishes which was available in the local markets of the Mysore city. So economic importance of the fishes. So fish constitutes an essential part of the human diet because of its easy digestibility and high nutritional value. It is also known as the most important source of the high quality of the protein for the especially for the humans. So modern fish farming with the high stocking densities, which provides an ideal conditions for the invasion of the pathogens, which may be of bacteria, viruses, or and the metazoan parasites. So infection by this disease, which reduces the condition and also the survival of the fish, which affects the economic losses, which also causes the economic losses in the fish. Cultured carbs are susceptible to the various kinds of disease, which may be of bacteria, viral, or and the parasitic. So we go on to the definition of the parasite. Parasite is an organism which lives on the host and which gets the food from the expense of its host. There are two types of the parasites. Ectoparasites are those which is present on the surface of the fish. For example, the fish lice and also the anchor worms. The endoparasites are those which is present in the internal organs of the body, such as the digestive tract, body cavities, and other tissues. The examples of the endoparasites are the tapeworms, roundworms, and Canthocephalus, etc. So these parasites, which affects the growth of the fish survival, it also affects the reproductive rate and also which affects the marketability of the aquaculture and also the fisheries product. So these parasite, it causes us the disease in the humans when it accidentally ingested along with the raw or the undercooked fish. So coming to the methodology of our project work, so we have collected sample of about 35 fish samples were randomly collected from the different local fish markets during the study period from November 2021 to July 2022. Then the samples were transported immediately in a plastic bag to the laboratory of the JSS College for identification and examination of the parasites. This study was taken place every week for a period of the 9 months. So next, examination of the samples and the identification of the parasites. First of all, first the external surface of the fish sample was examined thoroughly with the help of the hand lens, and then the fish was dissected, and then the internal organs such as the liver, intestine, gallbladder, stomach was examined thoroughly. So after examining the external surface of the organ and the organ was dissected for the further examination, then a drop of the water was taken in a clean glass slide and a small amount of the sample material, that is intestine, liver, stomach, gallbladder, was added on it. The material was mixed with the glass lid and separated thinly or directly collected endoparasites from the external surface of the fish organ was taken in a clean dry glass slide and placed by placing a water on this slide. Then the prepared slide was then examined under the stereosome and also as well as the compound microscope. Then the photographs were taken by using the mobile camera. Then the ectoparasites and the endoparasites which was collected was then preserved in 1% formalin and 70% alcohol for the further use. So coming to on to the results, total 35 samples of the fish were collected from the local fish market and was processed for the isolation and identification for the different parasites. So we have identified 6 parasites which has been identified, of which 5 are endoparasites and 1 is ectoparasite, and other 2 parasites which was found and that were unidentified. So fish parasites have been of great concern. These parasites which was continuously infect the fish, which may lead to the reduction in the growth and also the increased susceptibility to the diseases. Majority of the fishes carry heavy burden of the parasitic infestation, which causes deterioration in the food value of the fish. So growth pattern is useful in monitoring the state of the health of the fish species. In the present study, the ectoparasites and the endoparasites was observed in the different fish samples. So this is the checklist of the endoparasites and the ectoparasites which was found in the food fishes available in the local markets of the Mysore city. Then these are the photographs of the parasite which was collected. The plate 1, the plate 2, and these are the unidentified parasites which was found. Then coming on to the general characteristics of the the parasites. So first of all, the roundworm, which belongs to the kingdom Animalia, phylum Nematoda, class Chromadoria, order Ascaridida, family Ascarididae, and genus Ascaris. So this Ascaris is a genus of the small parasitic nematode worm, which was also known as the small intestine worm. It has elongated body with a cylindrical shape. This roundworm is very small and it is microscopic, which can measures up to 1 meter long also. So this is transmitted by infected fecal matter or consuming animal tissue with the worm larvae. The next endoparasite is Diphyllobothrium, which belongs to the kingdom Animalia, phylum Platyhelminthes, class Cestoda, order Diphyllobothridae, family Diphyllobothridae, genus Diphyllobothrium. So this organism belongs to the phylum Platyhelminthes, which are also known as a flatworms. This is a genus of the fish tapeworm which can cause a disease called diphyllobothriasis in the humans through consumption of a raw or the undercooked fish. In the year 1952, Dunas was the first who recognizably described Diphyllobothrium. And this Diphyllobothrium is anapolytic, which means it
Your
sheds
slide
their
is not visible.
proglottids. Sir?
Your slide is not visible.
Sir, it is visible now?
Yes, yes, very much.
So next, the Diphyllobothrium is anapolytic, which means it sheds their proglottids after the proglottids when it is matured. The next organism is Norellica. So this Norellica belongs to the kingdom Animalia, phylum Arthropoda, class Malacostraca, order Isopoda, family Cymothoidae, and genus Norellica. So these as isopods occur very commonly as parasites in the food, especially in the food fishes. So these Norellica is the blood-sucking parasites. In the Greek word, iso means equal and the poda means food, that which means it has an equal food. Then the body structure of an isopod, it consisting of a cephalon, which means the head, a thorax region, which means a perion, with the 8 segments, which is called as a perionids, and an abdomen pleon with the 6 segment, which is called as a pleonids, some of which pleonids, some of which may be fused also. So these isopods, which penetrates the skin with their periopods, and then as well as with the mouthparts also, and then the tissue inhabiting forms may which maintain a small opening to the outside. Then next coming to the Gammarus. So this is also an endoparasite which belongs to the kingdom Animalia, phylum Arthropoda, class Malacostraca, order Amphipoda, family Gammaridae, and genus Gammarus. So these are the amphipods which comprise an order of Crustacea, which is a shrimp-like form, which contains mostly marine freshwater forms. So while some species are terrestrial and some species are parasites also. So these amphipods were first described in the South Wales, in the Australia, and then later on several Pacific islands. So compound eyes of the amphipods are sessile, which means it is present flat on the head with no eye stalk. So these eggs are the Gammarus eggs are deposited within a brood pouch on the underside of the adult female amphipod's body. Then the next organism, the endoparasite, is an Acanthocephalus. So this Acanthocephalus belongs to the kingdom Animalia, phylum Acanthocephala, class Paleacanthocephala, order Echinoorchidae, family Echinoorchidae, genus Acanthocephalus. So in the Greek word, acantho means prickle and cephala means head. So Coel-Ruther was one of the scientists who recorded the Acanthocephalon from the intestine of the fish as a parasite and proposed the name Acanthocephalus in the year 1771. So the common name is thorny-headed worm because of the presence of the spines on the proboscis region there. So these Acanthocephalus are unsegmented worm-like and it is parasitic as Helminthes. Body is divisible into trunk and also the retractile proboscis there. So the proboscis region bears several rings of the spines that they use to attach themselves on especially on the intestinal walls of the fish or their host digestive system. The next organism which was found is the ectoparasite, which is called Orgulus species, which is commonly known as the fish lice. So this belongs to the kingdom Animalia, phylum Arthropoda, class Maxillopoda, order Orgulida, family Orgulidae, and genus Orgulus. So this is commonly known as a fish lice. It is dorsoventrally flattened. It has a specialized structure called stylate, which is located in the front of the mouth tube. So this is especially used to pierce the host and help the parasites to ingest their body fluids. So it has a direct life cycle with no intermediate host. So these fish lice has two prominent suckers, one located on each side of the mouth and stylate, which is especially used to attach to the fish. So conclusion, to conclude on the parasites of the food fishes available in the local markets of the Mysore city, parasites were recovered from 35 fish samples, which includes nematodes, cestodes, acanthocephalans, and also the crustacean parasites. It could be concluded that food fishes of the local markets of Mysore city, which are infested with internal as well as external parasites that could pose public health concern to fish consumers who consumes the raw or the undercooked fish. Fishes from the local markets of the Mysore city should be properly cooked before consumption to avoid any health risk due to some parasites. So thank you. These are my references.
Thank you so much. Please make to try explain this thing, not read the slides. This is the advice for you. Thank you. And next participant is Saloni Kamboj. Next participant, Saloni Kamboj.
Yes, sir. Yes, sir.
Please proceed.
Hello.
Yes, please proceed.
Hello.
Yes, please proceed.
Yes, sir.
Next participant, Serene Fatima Rajvi, please be ready and contact with Miss Karisma
Sir, my
or Miss Bhavna.
presentation is visible.
Yes, visible.
Okay, sir.
Miss Serene Fatima Rizvi, please contact Karisma ma'am or Bhavna ma'am to present next.
Okay,
Sir,
sir.
is my presentation is visible?
Yes, visible, very much visible. Please proceed.
Good afternoon to all of you. Today I am here to present my presentation on the topic of the source of high levels of metals and ions in drinking water and its effect on human health in third international conference on recent trend in life science and biotechnology under the supervision of Dr. Nirankar Singh, Associate Professor in M. Maharishi Markandeshwar University. These are the contents: introduction, selected parameter, water quality standard, source and harmful effect of the selected elements in drinking water, method to remove the metals and introduction. The three essentials of the life, air, water, and the food. Water occupy the second position in order of the priority, not only for human beings, but also for plants and animals. Many minerals are present in water. Some are present in excess quantity. If the values of these minerals are lies above the WHO and BIS standard, then it is not good for health. In this presentation, we are discuss about the metals and iron in drinking water and their sources and its effects on human health, and some methods to remove the heavy metals from the drinking water that affects the human health. This picture shows the metal and associated health risks. Heavy metals can cause lung cancer, stomach cancer, cornea heart disease, brain damage, physiochemical, neurological disturbance, etc. These are the selected parameters for the discussion. First is nickel, lead, selenium, copper, zinc, magnesium, iron, fluoride, and chloride. First is lead. Lead is a naturally occurring bluish green metal present in the small amount in the earth crust. Now, the sources of the lead in the drinking water, paint, soil, drinking water, etc. Harmful effect of the lead. When the lead are present in the drinking water, then it may cause many problems like lower IQ level, decreased ability to pay attention, unperformance at school, etc. And when the toxicity of the lead. When the lead is present in the drinking water in the excess amount, the greatest it may cause high kidney and nerve system in both children and adults, high blood pressure, joint and muscle pain, difficulties with memories or concentration, headache, mood disorder, etc. Now, the second element, second metal is nickel. Nickel is also an micronutrient essential for proper functioning of the human body. This metal makes its way to the human body through respiratory tract, digestive system, and the skin. Sources of the nickel in the drinking water. The primary source of the nickel in the drinking water is leaching from metals in contact with drinking water, such as pipes and fitting. Nickel may also be present in some groundwater as a consequence of dissolution from nickels or bearing rocks. Toxicity of the nickel. In some quantities, nickel is an essential, but when it is uptake too high, then it can be dangerous to human health. Nickel compounds in the body through chronic exposure may be responsible for a variety of adverse effect on human being. Nickel affect lung, fibrosis, kidney, cardiovascular disease, and cancers of respiratory tract. Now, the third element metals is selenium. Selenium is a metal found in a natural deposits or ore containing other elements. At typical groundwater pH values is 7.0 to 9.5. Only anionic form of selenium is selenium 4 or selenium 6 acid are found. Sources of the selenium in the drinking water can come from discharge from mines, natural way deposit discharge from refineries, or from agriculture runoff, leaching natural selenium compounds from dry undeveloped land. Pictures of the harmful, these are the pictures of harmful effect of selenium. These pictures shows if the selenium is present in drinking water, then in excess amount according to WHO and BIS standard, then it may cause many health effects like diarrhea, fingernails, hair loss, itching of skin, nausea, vomiting, etc. Iron. Iron is a mineral. Most of the iron in the body is found in the hemoglobin or red blood cells in the myoglobin. Iron is needed for transporting oxygen and carbon dioxide. Sources of the iron in drinking water. Iron exists naturally in river, lakes, and underground water. It may also be released to water from natural deposit, industrial waste, refinings of ores, etc. Iron toxicity. Excess iron can damage to the gastrointestinal system. Symptoms of iron toxicity include nausea, vomiting, diarrhea, and stomach pain. Over time, iron can accumulate in the organs and cause fatal damage to the liver or brain. Copper. Copper is a metal that occurs naturally and is used to make many products, including parts for plumbing systems. Sources of copper in the drinking water. Water absorbs copper as it leaves from pumping pipes, fitting, brass, etc. Coppers can get in drinking water as water passes through your household plumbing system. Harmful effect of selenium. Selenium is also an element which is present in the drinking water. When it is present in excess amount, then it may cause headache, diarrhea, blood warp, like the effect, etc. Zinc. Zinc is a mineral that is an essential trace element present in the water because very small amounts of the zinc are necessary for human health. It is also used for boosting of the immune system. Sources of the mineral in the drinking water. The main man-made release of zinc to the environment and water are from metal production process, from industrial combustion of coal, from waste and from worn rubber tires on vehicles. Small amounts are also released to the groundwater from the earth crust. Toxicity of the zinc. Zinc is an important cofactor in the body and it is essential for normal function. However, increased level of the zinc can become toxic. There are the three types of the exposure that can lead to toxicity: inhalation, oral, dermal. Most cases are acute toxicity, so treatment plants and minerals such as chelation therapy or prescribed medication, etc. Fluoride is found. It is an ion. It is found in the drinking water. It is also produced synthetically or used in drinking water, toothpaste, mouthwash, and various chemical products. Fluoride comes from fluorine, which is a common, natural, and abundant element. Adding fluoride to the water supply reducing the incidence of the tooth decay. Fluoride protects teeth from decay by demineralization and remineralization. Sources of the fluoride in the drinking water. Fluoride is a mineral that occurs naturally and it is released from rocks and soils. Almost all the water, some fluorides, but usually not enough to prevent tooth decay. Fluoride toxicity. When the fluoride are present in water in excess amount, then it may cause many health effects like skin problem, acne, cardiovascular problem, including high blood pressure, etc. Thyroid. Now the last one is chloride. At the concentration found in the drinking water, chlorine is non-toxic to humans. Many municipalities and add chlorine to their water to help kill harmful organisms such as virus, bacteria, etc. Sources of the chloride in the drinking water: agriculture, runoff, oil well waste, wastewater from industries, etc. Toxicity: chlorides can corrode metals and affect the taste of food products. Therefore, water that is used in industry or proceed or for many uses recommended maximum chloride level. Chloride increase the electrical conductivity of the water, thus increase its porosity in metals pipes. Chloride react with metals ion to form soluble salts, thus increase levels of the metals in the drinking water. Now, the methods are to remove the metals and ions from the drinking water. There are many methods to remove the metals and ions from the drinking water. They are like reverse osmosis, ion exchange. To remove heavy metal from the wastewater, many conventional techniques are used. Metals are removed from solution by increasing the pH of the effluent, converting the soluble metal to an insoluble form. Conclusion: when the metals and ions are present in the drinking water in the excess amount, then it is very dangerous for human health. These are the references. Thank you.
Thank you. Next participant, Sarin Fatima Rijvi.
Yes, sir. Am I audible?
Yes, audible.
Yes, sir. So, a very good evening, everyone. Myself, Shireen Fatima Rizvi. I'm a PhD scholar from Integral University. I'll be presenting a poster themed as valorization of chicken feather hydrolysate as a solid substrate for extraction of microbial PHAs. Okay, so all of us here are very much aware about.
Ma'am, your slide is not visible.
Sir, ask Bhavna ma'am to present it. She's presenting on my behalf.
Miss Bhavna, please present.
Is my screen visible?
Yes, visible very much.
Okay.
So we all are very much aware about the problem that plastic is causing to our environment. This work is an attempt to solve that problem to some extent. PHAs here are polyhydroxyalkanoates. These are polymers which have qualities very much, physical qualities very much similar to that of plastic. And these can and provided with a plus point that these are biodegradable, fully biodegradable and biocompatible. So they can be used as plastic material, as packaging material, as well as in implants and for in human bodies and for surgeries and all. So the being so good, the only problem associated with PHA production is the cost. microorganisms accumulate PHA when they are grown under nutritively stressed environment. They accumulate it as a stress mechanism. So this nutritive stress has to be, when incorporated artificially, has to we have to give a surplus amount of carbon source to the microbial culture in order to accumulate those huge amount of PHA. Uh, uh, this is the basic problem, the surplus of carbon. How we give it? In laboratories, we do by giving glucose, huge amount of glucose to the culture. But this makes the production cost very high of the pH. So here I'll be using chicken feather hydrolysate in my culture media for the production of PHA. So I will directly come to the methodology of my work. First, I have isolated soil samples from two different sites from Lucknow. I have screened them for PHA positive colonies. Then I have done screening of those samples. Screening was done using two dyes, Sudan black and Nile red. These two dyes are you can see the literature. These two dyes have always been used for PHA detection. Then the third step of my methodology is preparation of hydrolysate. It has been the most difficult step, I must say. I have collected the discarded chicken feathers from butcher shops, and then I washed it thoroughly using distilled water, detergent, ethanol, everything, and followed by drying. I prepared the powder, which was used for preparing the hydrolysate. Secondly, I use that hydrolysate for the preparation of media, in which I inoculated the strains which I found positive, which I isolated, which were positive from my screening step. So after the inoculation, they were incubated for on 37 degrees Celsius for 48 hours, and after that, I have extracted my polymer. So the polymer extraction was done using solvent method. I have used chloroform for that, and these are my results. So I collected soil from a slaughterhouse and a bird farm. I selected such sites because I wanted my microorganism to be able to utilize and be utilized I wanted my microorganism to be able to utilize the animal waste. So these sites were ideal for such microbes. Total colonies, the number of total colonies selected here were actually random. I selected these colonies randomly, and out of these I found from the first side I found 32 positive strains, and in the second slide from the second side I found 18 positive isolates. So these are the pictures. First one, the figure 1 shows picture for PHA positive strains using Sudan black staining. Sudan black is basically a dye which is lipophilic, lipid loving, and PHAs are very much similar to lipids. So these are the screening images. And second is Nile red. Nile red is a fluorescent dye. We use it we incorporate it in the media, and then after the colonies are grown, when you will see these plates in the UV chamber or something, the positive colonies will glow. So you can see some of the picture is not very clear though, but you can see some of the colonies are glowing like stars, and other are quite not glowing well. So these were the positive ones. And the third figure shows the PHA extracted after the solvent extraction method. The conclusion of the study is I have to do a lot of work. I have to take it a lot of
I have to do a lot of work further. I will be studying the strain by bacterial strains which I found positive very thoroughly, as they will help me in optimizing the yield of the polymer. And I'll be using different other waste for the production of PHA, and let's see what hopefully I'll find something good. So that's all. These are the references. That's all. Thank you so much. Any questions? Any questions?
Next participant, Richa Sukla. Next participant, Richa Sukla.
Yes, sir. Am I audible?
Yes, please proceed.
Okay, sir.